Bismillahirrahmanirrahim. Bismillahirrahmanirrahim and Assalamu alaikum and good day, ladies and gentlemen. I'm Hazima Wazam and I welcome you to online course on the series, fourth online course in the among the series of our online courses on biotechnology. This course is titled as Micropropagation of Quality Banana Plant for Commercial Purpose. This course is jointly organized by Comstech Secretariat and in collaboration with International Center for Chemical and Biological Sciences, Pakistan, and Pakistan Biotechnology Information Center, Karachi chapter. We are very grateful for our international experts and the local expert who have arranged this online course. And we are also very grateful to our participants who are joining us from different corners of the world and in the OIC countries. To officially start, I request Professor Dr. Iqbal Chaudhry for his welcome address and introduction. Welcome to the speaker.
Bismillahirrahmanirrahim. Assalamu alaikum. Good morning and good evening. I know that people from different time zones are joining this very important capacity building workshop on a topic which is very relevant, and that is agriculture and modern technologies. So I'm absolutely delighted that Pakistan Biotechnology Information Center along with Comstech are jointly organizing this series of events, and as I understand, this is the third one. And it is focusing on banana. Banana is considered to be a cash crop. It's also a staple food for many countries in West Africa and Central Africa, as well as in South America. Certainly a very important crop, which brings lots of revenues to many countries of the world. Now, banana is also one of those crops which are prone to diseases. And we have seen in many countries of the world that banana was actually wiped out because of very different kinds of diseases, including viral disease. Comstech has a major program in agriculture. We are working with the Islamic Organization of Food Security on different aspects of food security in agriculture, and with other partners as well. So this workshop is very important because this is going to develop your capacity in plant biotechnology, largely micropropagation by using tissue culture. Tissue culture, technological development, it is easy to perform. It's not very high technology stuff. That is something which even the farmers can do with clean air and skill. Tissue culture provides you a virus-free germplasm. It provides you plants which are healthy to begin with, and as a result of it, you're not really facing problems which can lead to product quality compromise. So this workshop is certainly very important, and I hope that you would be able to benefit from the deliberation, the innovation laboratories. You would be visiting online different labs. You would also be visiting tissue culture laboratories, and as well as various research labs. And you would then understand, hopefully, that this technology is certainly a very important one, which can be applied in any country of the world without much of a hassle. So kindly concentrate on developing your skill, ask questions, enter into the chat box, also in discussion rooms, and make sure that any question arise of the deliberation and lectures could also be responded in a timely manner. I would like to thank my colleague, Professor Dr. Samar Yusuf of PABIC, Pakistan Biotechnology Information Center, my colleague Hazima Mazum from Comstech for organizing this very useful and important event. And I wish you all the best. I hope that this event would be useful for all of you, and hopefully to see you soon, inshallah, in next of such event with PABIC. Thank you. Wassalamu alaikum.
Thank you very much, sir, for your kind words. And with this, we will begin our technical session for this online course. Our first lecture is by Professor Dr. Paul Chegg. Professor Dr. Paul Chegg is as an associate with the International Services for Acquisition of Agri-Biotech Applications, ISA, African chapter, with the role of information and knowledge materials development. Paul obtained his PhD degree in crop and horticulture sciences from Hungarian University of Agriculture and Life Sciences after successfully defending his doctoral dissertation titled Improvement of Somatic Embryogenesis and Androgenesis System for Sorghum towards production of double haploid lines in the selected Hungarian varieties. We are very grateful to Professor Dr. Polchak for his time and for accepting our invitation. His title for the talk today is Basic Plant Tissue Culture Techniques, the Role of Androgenesis and Embryogenesis in Attainment of SDG 2. With this, I welcome Dr. Polchak for his talk.
Thank you, Kazima, and thank you, everyone, for your kind welcome. I hope you can see my slide.
Yes.
So, thank you, Professor Chaudhry, Professor Samir, and Kazima for the invitation to give this talk. I will do so in about 35 minutes. The first part will highlight on the world situation today, and then I will touch on the basics of tissue culture, then give you 2 case studies with the sorghum, my crop of interest, and then leave you with why we should do tissue culture for bananas. And I'm sure all what we shall learn in the first part, you will practically see in the practical session. So my topic is titled Basic Plant Tissue Culture Techniques and the Role of Androgenesis and Embryogenesis in Attainment of SDG 2, as Kazima has mentioned. Sorry, I don't know why. So our world today. So the 2030 Agenda for Sustainable Development puts forward a transformational vision, recognizing that our world is changing, bringing with it new challenges that must be overcome if we are to live in a world without hunger, food insecurity, malnutrition in any of its form. So the world population is continuing to grow, and majority of the population now actually live in the urban areas. And the world economy is not growing as much as expected, with negative growth in most parts of the world in the 2020s. There is conflict and instability, and as you can probably hear recently in my region, Ethiopia, unfortunately, climate change and increasing climate variability and extremes are affecting agricultural productivity, food production, and natural resources. And all of this lead to major shifts in the way in which food is produced, distributed, and consumed worldwide, and to new food security, nutrition, and health challenges. So more than 820 million people are still hungry today. And this underscores the challenge, the immense challenge of achieving the zero hunger target by 2030. Hunger is rising in almost all subregions of Africa, especially, and to a lesser extent in Latin America and Western Asia. In Africa, in the last decades, only Western Africa has managed to reduce the number of undernourished people. And it is sad, sorry, it's sad to note that 9 out of malnourished children are from 9 out of 10 out of malnourished children are from Africa and Asia. So how do we end hunger, achieve food security, and improved nutrition, and promote sustainable agriculture? The game changer has to be agricultural systems that will have to adopt sustainable intensification strategies with diminishing resources, but with increased productivity in the same with the same resources. The agriculture sector must adopt strategies that are attractive to the youth, unlike the current situation where the sector is characterized by the aged in most parts of Africa and Asia. The sector must adopt modern technologies, such as agricultural biotechnology, to be combined with other tools, including plant breeding, conservation tillage, integrated pest management, IPM, sustainable resource management, variety selection, organic farming, and utilization of indigenous knowledge. There is clear demand for new products to mitigate climate change impacts, such as drought and emerging challenges such as maize diseases and new pests. And new biotech products are required to meet food and nutritional requirements of an increasing population. So what is the role of tissue culture in this? So just to go through the basics of tissue culture is the in vitro aseptic culture of cells, tissues, organs, or whole plants. In this part, I will go slowly so that because you are likely to meet these terms in the practical session. So the in vitro aseptic, in vitro means in a contained space or in glasses. The aseptic means clean, free of contamination. And these cells, tissues, and organs are what we collectively call explants. Under controlled nutritional and environmental conditions, often to produce the clones of plants. So the resultant clones are true to type. That is, they are similar to the parent plant. And this technique depends mainly on the concept of totipotency. This means that the plant cells have the ability to, to express the full genome of by cell division. That is, we shall get the whole plant from a single cell, but also on the plasticity of cells. That is, capacity of the cells to alter their metabolism or their developmental pathway. So these control conditions provide a conducive environment for cell growth and multiplication. And the conditions are adequate supply of nutrients. The pH of the medium is important. Adequate temperature or optimum temperature, and proper gaseous and liquid environment, which are realized using an appropriate culture medium. A culture medium consists mainly of inorganic and carbon and nutrient sources, what we normally call macro and micronutrients, organic supplements, and here we have vitamins, amino acids in some cases, and growth regulators such as auxins, cytokinins, gibberellins, and some gelling agents, depending on whether the medium is liquid or solid. Then we may have gelling agents such as agar or gerolite. So you may find differences in this. So the pH alongside plant growth regulators are important in that they control the plant growth or the developmental pathway of plants and the antioxidant activity on the medium. So the composition of the medium, particularly that of hormones and the nitrogen source, has profound effects on the response of the initial explant. So plant growth regulators play an essential role in determining the developmental pathway, as I have already mentioned. And for instance, if we look at auxin and cytokinin concentration in a medium, when we have high auxin concentration and low cytokinin, then we are likely to end up with root formation. If we have low auxin and high cytokinin, then we are likely to have shoot proliferation. And the appearance of either roots or auxins, roots or shoots in a callus, which is the mass of cells when the hormones are balanced, then can indicate in some occasions your success in medium or in tissue culture. So gibberellins play a critical role in cell elongation and plant growth. So some of the common medium you will hear include the MS medium, Muller-Shigen Scrooge medium, 2N6 medium, 192 medium, and so on, depending on the composition of the main elements. So how are these applicable? So the first, or probably what is very important in banana production, will be micropropagation and meristem culture. And this is for reasons I will show you later. But we also may use tissue culture for micro manipulation or breeding, micro breeding. Then in cryopreservation, when we have to preserve germplasm or seed material over a long period of time, then we need very low temperature and certainly medium. Then anther-culture, where we have to alter the developmental pathway of pollen cells or pollen cells to, from sporophytic to somatic. And then somaclonal or gametoclonal variant selections, then transgene selections, where you select the successful transgenes. This is still done in tissue culture. Biofarming, where we have to harvest important elements from cells. Then propagation of hard to propagate plants, and then protoplast culture, which can be applied in the recent technologies such as genome editing and plant breeding. So we can actually call tissue culture a downstream technology, when you will still need it after doing genome editing, so that you can produce the desired plant materials. In that case, so tissue culture is indispensable, and we have to learn this art. However simple as Professor Chaudhry has said it is, we need it. We need it even in these new breeding technologies. So some of the challenges we experience in tissue culture include maintenance of an aseptic condition, and if scientists in the room will agree with me that this is one of the greatest challenge we experience. and getting an aseptic condition will ultimately, aseptic condition of both the explants and the working environment will ultimately determine your success in tissue culture. Then recalcitrancy, this is sometimes, the materials are, the explants are not successful in tissue culture, and this is contributed to by the genotype dependence of the material you are using, and the lack of, reproducible protocols. Some protocols, cannot be copied or transferable from one lab to another, much as you try. I don't know whether it is because of the transparency in science. Sometimes I understand some of the protocols available may not be very accurate, or something else, but this calls for optimization that you cannot, you still need to try in your own lab these protocols that are published online. So another challenge we experience is overproduction of phenolics, and this is very common with the bananas and also with my plants of interest. It's mostly contributed to by death of cells in culture, and controlling this also determines your success in tissue culture. And this is a limiting factor even in adoption of new breeding technologies for some of these crops. Then appearance of albino plantlets. This is not what you really want to see when you do your tissue culture. You don't want to see the picture you can see on your right. You want to see a green plant. So when this happens, then you are not very successful. And this happens. This is something you can expect in tissue culture. And then we need skilled manpower to, we can, much as it's simple to do, you have to be trained. We don't have, there's a scientific process, and scientific process requires efficiency as well as accuracy. And yeah, we need skilled manpower. So my main message for this part of tissue culture is that progress in tissue culture technology has been achieved for many crops and in many parts of the world through empirical, detailed and time and cost-consuming testing of protocols. That these protocols have to be optimized. And consequently, success over the last few decades has been proportional to the number of dedicated laboratories involved. So we have to have, we have to be deliberate about having laboratories dedicated to a particular crop and a particular procedure in tissue culture. So yeah, so I will go to my case studies. I promised to give you 2 case studies, and one was both are projects that I was involved in somewhere in Hungary while doing my during my doctoral studies. And one is on optimizing tissue culture protocols for sorghum somatic embryogenesis. Sorghum is one of the most recalcitrant cereals. That is, it's difficult to culture, and protocols that are available are not reproducible. So we embarked in searching for a workable protocol for somatic embryogenesis. The importance of this is that with a workable protocol, you can even take sorghum to there could be projects designed for sorghum in new breeding technologies. If you don't have these protocols, then you are not likely to take, this crop may not benefit from this, yet it's a food security crop, just like banana. It is a staple crop in many parts of Africa and I'm sure in Asia as well. So we used 10 genotypes from a cereal research company in Hungary. And so the first thing is making sure that the seeds are free. The seeds were, the explant used was seed to start the process, and they had to be disinfected. And how this was done, the seeds would be washed first with 70% ethanol. And then after that, they would be washed alongside a shaker in 4% sodium hypochlorite with some drops, 2 drops of tin 20. And then rinsed three rinses, of course, in a clean bench, which I'm sure you will see all this in the coming practical session. And then we used the basic MS medium. When I say basic, I mean one that does not have plant growth regulator, does not have hormones. The point of this was to get mesocotyls, which were the real explants that were used. We sought to get the actively dividing parts, and the experiments were set in RCBD, and incubation was done for 4 weeks in dark thermostat at 28 degrees. So the medium types involved for this trial included half MS medium, and constituents of the medium are given. Then half 192 medium, then half NS6, then half B5 medium, then a medium. So half MS medium, and modified with amino acids, proline and asparagine, as well as with potassium hydrogen phosphate that had to be filter sterilized, and then a control medium that is full MS, full strength MS, what you find published as MS medium, but then modified with the proline, with amino acids, proline, asparagine, and copper sulfate. So what was observed is that the half strength MS medium produced the most quality kali. The most quality kali, as you can see, medium E in the table on the left, produced the most quality kali. A quality kali is described as a kali that is yellow or white, and depending on the plant, and friable. That is, it has parts that are easily, you can detach. It's friable, a friable kali. If you get brown, it means that you do not overcome phenolics, and phenolics is what I showed you in the picture previously, where the medium is completely black. So, what we deduced from this is that half strength medium, okay, MS medium was the best, emerged as the best among them, among the medium tested, but then half strength of the MS gave more quality kali than full strength MS, the control medium F. The second part of The second case study that I wanted to show you is on androgenesis. And in this, we sought to produce doubled haploids. And you can see that these are completely very different sets of experiments, because the medium types and the explant types are completely different. So for this, you have to have the pollen as the the medium, the explant. So we used 28 germplasms here, sourced from grass houses as well as open field cultivation in both summers and winters. And the trick here is to obtain the material or the pollen or the anthers when the panicle, is just before the lateral expansion has caused the leaf sheath to split open. And sometimes you have to involve pretreatments, but in this case, as we tested, we found that pretreatment for sorghum was not so important as getting the panicles at the right stage. The essence of pretreatments, first of all, you may get materials from the field and pretreat them, have a temperature and light pretreatment, and this is basically for synchronizing the maturation of the explants. The other pretreatment is usually in culture, and this is to ensure that to change or to induce the change from sporophytic development to somatic development. So because you want the pollen, which is basically a gametophyte, to change its development pathway to somatic, to become a plant. So we tested various medium types. I don't want to go to the details of this. And what we emerged with as the best from this was a fecal saturated or fecal supplemented W14 MF medium. W14 MF medium is just the same as it's just one medium type, just like MS or NS, N6 or B5. So W14 MF supplemented with fecal. Fecal is a hydrophilic polysaccharide. This emerged as the best. So in this experiment, or in this type of tissue culture, you also have to, once you obtain doubled haploids, if you are successful, because for us we were able to obtain 5 lines that were haploids, you have to either wait for spontaneous diploidization, or you can do chromosome duplication through colicin. Colicin induced. I'm sure some of you have heard of colicin induction. Colicin is an anti-mitotic agent, and it helps to double the ploidy level of the haploids obtained. But in the case of sorghum, we saw that, or we arrived at the conclusion that spontaneous diploidization was sufficient enough and gave us sufficient fertility levels of up to 80.5%. So we really did not need to do colicin duplication. Again, some people have fear on colicin use, understandably. So I won't go to the politics of that. But for this crop, spontaneous deprotization was sufficient. So again, in this type of tissue culture, you have to eventually determine the ploidy of the materials you obtain, because the essence of doubled haploid is to get homogeneous homozygous populations in a shorter time than you would need to do breeding and achieve homozygosity with repeated back crossing. So this technology helps you to reduce the breeding time by probably 6 generations. So instead of doing 8 generations of back crossing to get homozygous populations, you do it in 2-3 generations, you have homozygous populations. So yes, for this you have to do ploidy determination, and this you can do either with flow cytometry or using markers. For our case, we used SSR markers. So those are my 2 case studies. I wanted to also highlight just before I come to this, just to say that the points I have mentioned will certainly be applicable in 1 or 2 of them will be applicable in banana tissue culture. I'm sure in banana tissue culture as well, we shall still have explants. We shall have to obtain explants. We shall have to do aseptic, we shall have to assure asepsis, that is, we shall have to be free of contamination in the working environment, and as well as on the explant itself. And we shall have to get the medium right. We shall have to get the medium right. And for this, we have to optimize. And then we shall have to get the hormone concentration right. And then after that, we shall have to, to ensure whether there will be pre-treatments or not. And then we shall have to have the induction conditions, or the induction, the routing conditions, right? We shall also have to have in the process after we obtained the plant, and we shall have to ensure that the plant that we obtain is true to type. It is what we expected to get. So these are commonalities in tissue culture. So for tissue culture of bananas, why do we have to do tissue culture of bananas? Because sometimes it is the only way to eliminate some diseases. If I mentioned about meristem culture, and meristem culture is very helpful in elimination of some very, let's call them persistent, or for lack of a better term, pathogens, such as those that cause black sigatoka. I'm sure there are banana experts here who we may add other pathogens to that list. And then through micropropagation, certainly we'll have clones. So we'll have relatively phenotypically uniform seed material. So the seed material that you start your plantation or your enterprise with, you are sure that they are phenotypically uniform. They are obtained from the same parent. Then certainly the elimination of these pests and diseases contributes to high yields, and fast maturity, because of course the plant is free from stressors and therefore can express itself fully. the genome can be expressed fully, and therefore you can enjoy the full potential of the genotype that you planted. And then tissue culture has the advantage of getting us or delivering to us high-yielding farmer preferred cultivars in a shorter time than of course we would do with the other methods of propagation for suckers and so on for bananas. So I end it here. I'm sure there are many questions, but more of the tissue culture of bananas is coming in the practical session. I'm sure Kazima has organized that. So I thank you so much for your attention. I hope I was not, I was clear, because of our geographical differences, probably my pronunciation of some words could have issues. So thank you so much.
Thank you very much, Professor Dr. Paul. It was a very nice presentation and very excellently put together. We have some questions for you from the participants. Should I read out the questions for you?
Yes, please.
Okay, so first question is from Mr. Mehdi Hassan Sohag. He's asking, in a real case scenario for the research laboratory in academia, it is very challenging to ensure the asepsis because multidisciplinary life sciences work. So his first question is basically, how do you think is the best possible strategy to reduce contamination? Okay, let me read out the question again for you. In a real scenario for research laboratory in academia, it is very challenging to ensure asepsis because multidisciplinary life sciences works can be optimum source of fungal and other concomitant particles are the source of contamination. How do you think is the best strategy to reduce contamination, number one? And number two, he's asking in terms of acclimatization.
Acclimatization.
Yeah,
Yes.
the response rate of the explant from the tissue culture for non-conventional crops is not satisfactory yet. What is your opinion about this?
Yeah. Can I answer those?
Yes.
Okay. Maybe Hassani on the possible strategies to reduce contamination. The first thing in tissue culture is to be aware that this is a challenge. Before you even begin your work, you must have this in mind that contamination happens, and contamination can be from the explant itself as well as the environment that you're working with or yourself. You can be the source of contamination. So the first thing is to ensure that you obtain a relatively clean material where you get the explant from. They should be physically clean to you. And then the second thing is to ensure that you have a strategy for ensuring the aseptic condition for the explant. And this you have to optimize as well, because sometimes what works for one crop or what works for one explant in a crop is not the same as the next. For instance, for sorghum seed, the dry seed, you have to start with ethanol treatment. But for anthers, panicles, you can't use the ethanol part first, because of course this is the ethanol can mess with your explant. So you have to you start with sodium hypochlorite immediately. You avoid the ethanol part. So you have to be very deliberate with your treatment remove contamination before you go to the clean bench working. Now, the clean bench has to be sterile, and you have to sterilize this. You have to optimize. You have to know this. You have to put this in mind as you start working. And the easiest way to sterilize, of course, is to use ethanol, and also to use the UV light on that is provided on the clean bench. The second thing is for you to ensure that you are not the source of contamination. Working outside the clean bench, your hands inside the clean bench, but your body and your clothes, of course, I'm assuming that you are in the right PPE, you have your lab coats and gloves, but also, stay clear of the clean bench as you're working, so that, only your hands are in the clean bench, because contamination is one part of tissue culture that can really slow you down. And also, please ensure that you are not compromising. You don't compromise contamination. If you detect any contamination, then it means that you discard the material. Another source of contamination would be the plates, the battery plates that you are using. Sometimes glass or plastic battery plates can be the reason why your medium is contaminated. So I don't know whether I have answered this, but please also, once you are done with the operation on the clean bench, ensure that you seal properly to keep off, during the induction process and the routing, you keep off contaminants from your explants. So on the acclimatization response rate, first of all, if you have already obtained a callus or cali, and you have already routed this, you have already obtained plantlets, and now you are at acclimatization stage. This should be, as a scientist, this should be the less challenging part for you. And I say this because you have some level of control on adaptation of the crop of the plants in greenhouses. You can regulate the amount of light, the amount of humidity necessary during the acclimatization process. Again, this needs optimization, but it's the easier part. I think the harder part is to obtain the plantlets. I hope that answers the two questions.
Thank you very much, Jug. We have another question from Dr. Saifullah. He's asking at what stage you choose plant parts for culturing. For example, in Panama disease start at early stages, but the symptoms appear at the later stages. So how you screen plants for culturing?
Sorry, come up again, Kazima.
So the question is, at what stage you choose plant parts for culturing? For example, in Panama disease, start at early stages, but the symptoms appear at the later stages. So how you can screen plants for culturing? Okay,
so as I said, for banana, elimination of diseases, the most effective procedure is the meristem culture. And the advantage of meristem is, as many plant scientists will agree with me, is that they are free of pathogens. And so regardless of the stage of the plant, or because you might want to save the plant when already infection has occurred. So long as you can obtain a meristem, then then you are, you stand a chance of rescuing the plant from the infection, the viral infection or the bacterial infection.
Thank you very much, Chuck. So here is another question from Mr. Khizar Nazir. He's asking how to control endophytic contaminations.
So endophytic contaminations, I believe you're talking about what happens outside the cell or outside. I don't know whether you mean endophytic contamination in culture or while the plant is in vitro or during the acclimatization stage or already in the field. Of course, our aim is to get clean material eventually. So, what you're working aseptically is one way to assure that you keep away external contamination, but also in field, please ensure that the field you are planting the material into is not where you just removed the contaminated plant material. So you have to ensure that you are starting clean, you are starting on a clean plate. So the field that you are planting your new material should, at least be free of the vegetative materials of the former plant. So that's one way of keeping the new contaminants away from your plants.
Thank you very much. Check, here is another question. How to ensure genetic stability in tissue cultured plants?
Genetic stability, first of all, in tissue culture, we may have variants. We may have variants, but and this can easily be detected, first of all, phenotypically, and also through molecular mechanisms such as PCR. But again, your aim in tissue culture is to get phenotypically uniform plantlets. So if you get variants, it's advantageous if you are a plant breeder, and a variant can be helpful or maybe cannot be helpful depending on your aim. But in most cases, we are interested in the phenotypically uniform plant material, or also the homozygous, attainment if you are talking about haploids and double haploids.
Thank you very much, Jeg. We have another question from Mr. Hamza Dar. He's asking that in our lab in China, we are doing pumpkin transformation, but face a lot of fungal contamination. Even we sterilize the seeds with ethanol and sodium hypochlorite. What is your suggestion in this regard?
Okay, thank you so much for that question. I would be interested to know the stage of the stage at which you are using the seed material. If it is completely dry or it's still wet. Sometimes when it is dry, some seeds have endophytic. They have some pathogens under the seed coat. So, and these are hard to reach parts. So of course they reduce your efficiency, but if you can, it helps. I have tried this practically. It helps to remove the seed coat sometimes, so that you can reach them below
Online
the seed
course.
coat.
Sorry.
Yeah, that is one way of ensuring. So depending on the stage at which you get these seeds. Again, it also depends on how long the seeds have stayed post-harvest. So sometimes you may get contamination during harvest, during storage of seeds, and seeds with a long keep post-harvest are likely to be problematic in ensuring that they are free of pathogens. So this is something you have to keep on trying. You have to keep on trying until you get it right. But if you are dealing with dry seeds, then if they are completely dry, you may try to remove the seed coat and work with the seed without the yeast seed. Yes.
Right. So here is another question. Which synthetic hormone in case of banana culture are most useful, and what concentration is more useful?
Oh, that is a very good question. And I really wanted to bring you very specific banana tissue culture, but unfortunately, I was not able to obtain these materials from the labs that are using them. But I am sure that this is something that needs to be optimized. Just try the available, what you have. Start with what you have. If you have 2,4-D for auxins, or if you have whatever cytokinins you may have, please try to optimize as much as you can, because sometimes I really wanted to bring you very specific protocols, but this was not possible on my part.
Paul,
Th-
sorry, Khazima, let me add one thing regarding this question, that many of the answers regarding the concentration of various nutrients or the growth hormones and something like that has already been provided in the manual, which we have already sent to the all registered participants. So you can also take this information from that manual.
Yes, exactly. And also I have, posted this manual in the chat box as well. If anybody has not received it in their email due to any reason, they can download this from the chat box as well. Dr. Cheng, we have last question for you from Mr. Raza Yasin. In how many crops we can use same technique we discussed here for banana and sorghum?
Sorry, again, please.
In how many crops we can use same technique we discussed here for banana and sorghum?
In how many crops?
Yeah.
Cereals, first of all, you can optimize a lot of what I discussed for sorghum with several cereal crops, such as wheat and wheat, barley, and so on. But then some of these crops have, their protocols are reproducible enough, so they don't have as much challenge as sorghum has, and as banana would have. Sorghum and banana are very close because of the challenge of phenotypes, phenolic exudation, or what we call lithobrowning. Lithobrowning causes some of the cells to dry or to die in culture, and this is very challenging, and you have to optimize each time. So I would suggest that once you obtain a workable protocol, one that you think is giving you some positivity, you try to optimize with that. But for what I presented, I'm sure it works because it worked for me. So, but also it depends on how accurate you are with the plant growth regulators, and also with amino acid modifications of the main medium type.
Thank you very much, Dr. Cheng. With this, I request Dr. Samar if she would like to say a few words about Professor Dr. Cheng in his talk today.
So thank you so much, Paul. for sparing your precious time with our audience. And the intensity of the questions actually demonstrated the interest of the participants in this specific area related to the tissue culture. Paul, if you allow us, we can drop your email address in our chat box so the participants can communicate you directly, because all the activities which are going on under the umbrella of the comstech and other organizations, our main purpose is to connect these scientists all over the country so they can interact with each other and they can share their problems, and with the emphasis to get some answers with their troubleshooting. So could you kindly allow us to drop your email ID in the chat box?
Yeah, thank you so much. In addition, I'm also available on LinkedIn and on Twitter. If you look for me, Paul Chege, you'll find me. So yeah, you are most welcome to contact me anytime.
Thank you so much, Paul. Khazina?
Yeah, Dr. Samar, should we move on to our practical session?
Yes, we can.
Okay, so thank you. After thanking Professor Dr. Chegg, we will move on to our practical session. This practical session will be delivered by our experts present at ICCBS and the team under Dr. Samar. And the topic for this practical session is tissue culture for cash crop, which is banana. With this, over to you, Dr. Samar.
Thank you so much. Let me start with the videos of each and every step of related to the banana tissue culture. And what I will request all of you to kindly drop your questions in the chat box. Our team is ready to answer you. So please see these videos. And if you have any question related to any step or the any technical aspect related to these steps, please feel free to drop your questions in the chat box. Okay, so we will start with our first video.
Tools and apparatus for tissue culture experiment. Apparatus required for tissue culture are forceps, scalpel, and blades, Petri plates with 2 filter paper. All the apparatus should be in spotlessly clean condition. Wrap scalpel, forceps, and blades in paper, brown paper, or newspaper. Wrap battery plates. Put cotton in the jars,
Hello,
which
Samar.
will be used to clean the laminar flow hood. Fill the bottles with deionized water, which will be used to sterilize Explant. These things are now ready to autoclave and will be used for tissue culture experiment later on. Media preparation. Preparation of stock solutions for MS media. Weigh desired amount of macronutrients in 1 liter of distilled water. one by one and store in refrigerator for further use. Weigh desired amount of micronutrients in 1 liter of distilled water, one by one, dissolve them properly, and store in refrigerator for further use. Prepare all the stock solution and store them for further use for the preparation of MS media. Preparation of MS media. Take distilled water from distillation unit. Distillation unit comprises of 2 units, deionized water and distilled water. Take 700 ml of distilled water for preparation of 1 liter of MS media. Turn on magnetic stirrer and put 700 ml of distilled water in a beaker on the stirrer, and also add a magnetic bead into the stirrer to dissolve the nutrients properly. All the stock solutions of MS media are now ready. First, add 50 ml of micronutrient stock solution into the MS media. Add 1 ml of micronutrient stock solution with the help of micropipetter. Add 1 ml of vitamin stock solution in 1 ml MS media. Add 5 ml of EDTA stock solution. Weigh 30 grams of sugar or sucrose for 1 liter of the media. Now add 30 grams of sugar into the media. Adjust pH of the media to 5.7 or 5.8 with 1 molar NaOH or 1 normal HCl. Make up the volume of the media in a measured cylinder. Make up the volume up to 1 liter with distilled water. Weigh 8 grams of agar, which we'll use to solidify the MS media. Add agar in MS media solution. Microwave the media to dissolve the agar completely for 10 minutes. Add phytohormones like auxin and cytokinins according to the plant requirements. Media is now ready to dispense in 250 ml glass bottles. Each jar contains measured quantity of 30 ml of MS media, close the caps of the jars carefully and tight. And proceed to the autoclave. Turn on the autoclave and open the lid. Place media jars carefully in the autoclave. Close the lid properly and check the outlet valves. Set autoclave program for 30 minutes at a temperature of 121 degrees Celsius and pressure of 15 psi. After autoclave, the media jars will be kept in growth room for 3 to 4 days to check any sign of contamination. In case you find any sign of contamination in your media jar, remove and discard that jar immediately. Contamination-free jars will be used for further experiment. Media jars can be stored at a temperature of 20 to 22 degrees Celsius. Process of removal of the contaminated jars from the growth room. First of all, collect all the contaminated jars and autoclave them. After autoclave, media should be collected in a plastic bag and proceed to discard. The jars are then washed and dried. Cleaning and preparation of laminar flow hood. Wash your hands properly with hand wash or soap before entering into the tissue culture lab. Before starting your experiment, you need to put on your personal protective equipment like lab coat, mask, pair of gloves, laminar flow hood is used to maintain the sterility of the cultures, and in order to maintain the sterility, we need to sterilize our hood first by exposing it with UV light for 15 minutes. Turn on UV light and cover the laminar flow hood as UV light is hazardous for health. Turn off UV light, remove the cover, turn on blower and light. Spray your hands with 70% ethanol before starting your experiment to disinfect them properly. Disinfect the surface of hood with 70% ethanol. Spray the surface of hood with 70% ethanol and wipe all the excess ethanol with autoclaved cotton. Disinfect all the objects with 70% ethanol you are planning to use during your experiment or need to bring into the hood, like media jars, autoclaved battery plates, sterilized tools, put the tools into the sterilizer and turn on the sterilizer. Laminar flow hood is now ready to use for tissue culture experiment. In order to turn off the hood, remove all the waste and used Petri plates and media jars. Turn off sterilizer. Disinfect the surface of hood with 70% ethanol. Remove all the excess ethanol with autoclaved cotton. Turn off switch, light and blower and cover the laminar flow hood.
Stage 1 of tissue culture is initiation. Substage, selection of explant. For tissue culture of banana, select a healthy, disease-free plant of banana from the field or greenhouse. Banana tissue culture can be initiated from shoot tips or sucker, but here we are using sucker as an explant. First of all, uproot the plant and remove soil from the plant. This is how the banana sucker looks like. Don't worry, we do not need to clean everything here as we are going to perform several steps to sterilize this material in the lab. First of all, we need to reduce the size of the sucker, something like that shown in the video. Basically, we need the innermost part and we can remove the extra layers around it one by one carefully. We can remove few more layers from the sides. We can also reduce the length of the sucker, but do not cut this point. Cut from the top and bottom of this point as this part is essential for tissue culture. So here we have the sucker of our desired size and we also saved the part that we need for tissue culture. Second substage of initiation is cleaning and sterilization of explant. As you have sucker of your desired size, now dip it in water or wash with running tap water. Now sterilize the washed plant material with 20% commercial bleach for at least 20 minutes. Cover the jar and gently shake it for 2 to 3 minutes. Expose the hood to UV light for at least 15 to 20 minutes and make sure that the hood is covered as UV light is hazardous for health. After 15 to 20 minutes, turn off UV light and remove the cover and then turn on blower and light. Place the jars with sterile water and plant material in bleach into the hood after spraying them with 70% ethanol. Spray the gloves with 70% ethanol and rub the hands to spread the ethanol prior to placing the hands into the chamber. Carefully open the jar with plant material and transfer the plant material into the sterile distilled water. Replace the lid of the jar and gently shake it for 2 to 3 minutes to completely remove the traces of bleach from the plant material, as we are sterilizing our plant material at this stage. Repeat this process 3 times to make sure that our plant is completely sterilized and free from all the traces of bleach. Remove the washed plant material from the jar with the help of sterile forceps and place it on a petri plate with 2 filter papers. Third substage of initiation is inoculation into the media. Now at this stage we continue cutting the sucker as we are going to reduce the size of the explant to 2 to 3 cm in length and 3 cm in width. Make sure to avoid any tissue that has been damaged by the bleach, which is apparent by its pale color, and cut all the tissues that you see that has been damaged by the bleach. You have to be very patient and cut little by little from the sides of the sucker and do not damage the innermost part, as this is the most crucial and essential part for tissue culture of banana. So here is a close-up of this part. Like you can see this central round part. This is the most important part that we need for tissue culture. As you can see, there are a few more layers around it, so we can cut few more layers around it to get the desired size of the explant. can see this line clearly, to reduce the size of the sucker, you can cut from the lower part of this line and from the upper part of this line as well to reduce the size of the sucker. And do not cut this line as you will damage your explant. Take a media jar and take a prepared plant section in sterile forceps. Place the prepared plant section into the media jar and press it a little so that it can get in touch with the media and get nutrients from the media. Transfer all the prepared plant materials into the media jars one by one. One thing which is very important, during the whole tissue culture procedure, you need to use sterile tools and do not use the hot tools as they can damage your plant. Replace the caps of the jars tightly and shift them into the growth room for further growth. After finishing your experiment, remove all the used jars and petri plates from the laminar flow hood and clean the surface of hood with 70% ethanol by spraying ethanol on the surface of the hood. Then wipe all the excess ethanol from the surface of hood with autoclaved cotton. These jars are then kept in the growth room with illuminated condition of at least 12 hours of light per day and at a temperature of 24 degrees plus minus 1 or 2 degrees for at least 28 days. Subculturing Stage 1. Basically, the purpose of this stage is multiplication of the plants. Objective of this stage is to increase or multiply the number of plants until you get your desired number of plants. Clean the surface of hood with 70% ethanol by spraying ethanol on the surface of the hood. And wipe all the excess ethanol from the surface of the hood with autoclaved cotton. Expose the hood to UV light for 15 minutes before starting your experiment and cover the hood as UV light is hazardous for health. After 15 minutes, turn off UV light and remove the cover and turn on blower and light to start your experiment. Spray the gloves with 70% ethanol and rub the hands to spread the ethanol before starting your experiment. Before taking the selected jars into the hood, spray them with 70% ethanol to disinfect them properly. The selected jars are now ready to be shifted into the freshly prepared media for multiplication purpose. Before starting your experiment, take the tools out of the sterilizer like scalpel and forceps. Arrange petri plates and jar. Always use cultures with no sign of contamination and use the media with the hormones according to the plant requirements. Open the jars carefully and take out the cultures from the jar with the help of sterile forceps, and do not use hot forceps as it can damage your plant. Remove phenolic, dried and dead tissues with the help of sterile sharp scalpel and forceps to avoid any contamination. Make a single sharp cut to avoid phenolic production. Remove all the leaves and keep lower stem part, as our aim is to multiply the plants at this stage. Now this part will be used for further multiplication of banana. Carefully transfer this part into the multiplication media with the help of sterile forceps and press it a little so that it can get in touch with the media to get nutrients. Here I mention again that do not use hot tools at any stage of tissue culture, as they can damage your plant. Repeat the same process for all the plants. As you can see that this black growth or production is due to phenolics, as banana produce little more phenolics. Avoid crowding of the cultures in one jar, as they need space and nutrients for their growth. Now cover the jar tightly with the cap. Label the jars with culture name, date, and type of media. Seal the jars with paraffin to minimize the chances of contamination. These jars are now ready to be shifted into the growth room for further growth. Turn off sterilizer after finishing your experiment. Repeat the same step as we did earlier. Clean the surface of hood with 70% ethanol by spraying ethanol on the surface of hood, and wipe out all the excess ethanol with autoclaved cotton. Turn off light and blower and all the other switches. Cover the hood. Now we will proceed to growth room with our cultures. These jars are then kept in growth room for at least 28 days under illuminated condition of at least 12 hours of light per day and at a temperature of 24 degrees plus minus 1 or 2 degrees. During this period, observe the cultures regularly to check the contamination. Keep visiting your growth room every 2 to 3 days to check the status of your cultures. You can see this white growth in the jar. It means it's contaminated and you have to discard it. Discard the contaminated jars immediately from your growth room and keep the contamination-free jars in your growth room for further growth.
Participants, if you have any questions, so our team is ready. Kindly start dropping your questions in the chat box. Thank you.
Subculture Stage 2. Again, the purpose of this stage is multiplication. Objective of this stage is same as stage 1. As we need more plants, so we are multiplying it again. Same steps will be followed as stage 1. Let's watch the video first and then I'll explain few things later on. The purpose of tissue culture is to produce number of plants from a single plant. Basically, banana can be subcultured up to 7 stages. At stage 1 of subculture, you will get 3 plants within 50 days. And at stage 2, you will get 12 plants within 75 to 80 days. And eventually, at stage 7, you will get 12,160 plants within 225 to 230 days. And you can stop subculturing at any stage like 4 and 5 as per your choice or as per your desired number of plants. Second stage of tissue culture is shooting. Objective of this stage is high frequency shoot regeneration. The growth and development of the tissues cultured in vitro are generally monitored by observing the cultures at regular interval. Visit your growth room to select the contamination-free jars for further procedure. Clean the surface of hood with 70% ethanol before starting your experiment. To clean the hood, spray 70% ethanol on the surface of the hood and wipe all the excess ethanol with autoclaved cotton. Turn on UV light for 15 minutes before starting your experiment and make sure that the hood is covered as UV light is hazardous for health. After 15 minutes, turn off UV light, remove the cover and turn on blower and light to initiate your experiment. Spray the gloves with 70% ethanol and hands rubbed together to spread the ethanol prior to placing your hands into the chamber before starting your experiment. For shooting purpose, same MS media with additional hormones required for shoot growth is used. Hormones required for shoot growth are NAA, that is naphthalene acetic acid, which is a synthetic auxin, and BAP, that is benzyl aminopurine, which is synthetic cytokinin. Take the selected jars into the hood. After spraying them with 70% ethanol, same precautions and full aseptic conditions have maintained throughout the whole process. Open the jar and carefully take the cultures out of the jar with the help of sterile forceps. Before transferring the cultures into fresh shooting media, remove all the dried, dead and phenolic parts with the help of sterile forceps and scalpel, and do not use hot tools as hot tools can damage the plant. Separate the plantlets from cluster as we cannot shift the whole cluster in one jar to avoid crowding in the jars. Take the media jar and open the lid and carefully transfer the separated plantlet into the shooting media jar with the help of sterile forceps, and do not use hot forceps or hot tools during the whole tissue culture process. Replace the lid tightly and avoid crowding of plantlets in one jar so that plants can get enough nutrients. Repeat the same process and transfer all the plantlets onto the shooting media to develop shoots. All the plantlets have been successfully shifted onto the shooting media. Seal the jars with paraffin to reduce the chances of contamination. Label the jars with culture name, date, and type of media. Shoots will be fully developed within almost 28 days. Now shift the jars into the growth room for further growth. The shoots will develop more quickly if the jars are placed under fluorescent light and they have provided at least 12 hours of light per day. Ensure that temperature doesn't go over 28 degrees and check the jars after every 2 to 3 days and discard those jars that shows any sign of contamination. Third stage of tissue culture is rooting. The objective of this stage is to prepare plantlets for transplanting by shifting them into rooting media to develop roots. First of all, from your growth room, you need to select the jars having well-developed shoots and are contamination-free to put them on rooting media to develop roots. Take selected jars into the hood after spraying them with 70% ethanol. As you can see, these jars are contamination-free and have well-developed shoots. Open the jar and carefully take out the shoots from the jar with the help of sterile forceps, and do not use hot forceps as it can damage the plant. After putting them on Petri plate, separate the individual shoots from the cluster before transferring them into the rooting media one by one, and also trim dried, dead, and phenolic parts from the developed shoots.
Take the jar with rooting media and transfer the developed shoots with the help of sterile forceps onto the shooting media to develop roots. And during the whole tissue procedure, ensure that your tools are not hot. Avoid crowding of shoots in one jar so that they can get enough nutrients. Replace the lid of jars tightly and also seal them with paraffin to avoid any kind of contamination. Label the jars with type of media, type of plant, and date. Incubate the cultures for 3 to 4 weeks to form roots. And when you observe the cultured plants with broad and expanded leaves and well-grown roots, you can transfer the plantlets to the soil. These plants are now ready to be shifted to the growth room for 3 to 4 weeks to develop roots. These jars are then kept in growth room for 3 to 4 weeks under illuminated condition of at least 12 hours of light per day and at a temperature of 24 degrees plus minus 1 or 2 degrees. Keep visiting your growth room after 2 to 3 days to check contamination status of your culture and discard those jars immediately from your growth room that shows any sign of contamination. Fourth stage of tissue culture is acclimatization. Objective of this stage is to shift well-rooted plantlets to the greenhouse. After 3 to 4 weeks, plants are well-rooted. All the healthy, disease-free plants are need to be shifted to the greenhouse in stepwise manner. It is a well-rooted plant and ready for acclimatization. Take out the jars from the growth room. Before taking them to the greenhouse, gently wash off all the agar or media attached to the tissue culture plants. Take out the plant carefully from the jar. Put them on a petri plate. Separate the plants from the cluster carefully without damaging the roots. Now wash the plants to get rid of the media attached to the roots. Gently wash all the plants without damaging the roots. Now we will proceed to the greenhouse for further process. Prior to planting tissue cultured banana plantlets into the soil, they need to be hardened or acclimatized to the external environment. This can be done by transferring plantlets onto the suitable media and exposing them to the partial sunlight under greenhouse conditions for a few days. Choose potting mix with good moisture holding and drainage characteristics. The media we used contains 80% of soil and 20% of cow dung, but you can use peat moss as well. Dip the washed and separated plants in the fungicide solution and then wash them with water to avoid any kind of fungal infections. At this stage, the plants are highly sensitive. Now transfer the plantlets on your selected media one by one. You can use any media at this stage according to your plant requirement. Place the plants in an area with 12 to 16 hours of light, either natural or artificial, but not the direct sunlight. Now cover the plants loosely with a plastic sheet. Plants still needs to be protected at this stage since they are not acclimated to the drier air compared to the moist environment of the jar.
So this was the end of the practical session. Let's have a look on the chat box. I hope our technical team has already responded many of your questions. The questions regarding internship, or if you wanted to have these tissue culture plantlets of banana, please do contact us on the two email addresses already provided to you by Farkhanda. So we will discuss and talk about for this later. Let me tell you one thing that the tissue culture section of the ICCBS has already been indulged in the internship programs. So if any one of you wanted to have the internship program, so you can contact us. Khazima, are you online? We will take a pause of, if there is
Yes, Dr. Summer, I'm online, and do we need to take some questions posted in the chat box?
Yeah, sure. I think many of the questions has already been answered, but you can check if there is any unanswered questions, so our team can answer that.
Okay, okay. So here is a question. Give me a minute, please. So they're asking, what kind of a fungicide are you using in your lab, generally speaking?
Assalamualaikum everyone. You can use different kind of fungicide, but mostly we are using mancozeb fungicide for this process.
Okay. So let me check if there are any questions. Definitely, your team, Dr. Summer, have answered most of the questions.
Yeah.
But I'm just taking a look at the chat box for the confirmation one last time. Dr. Samar, there are no more questions, but I would like to thank your team as well as you for this wonderful effort. And again, just to share with our participants, we will be sharing the manual that we have designed specifically for this online training course will be shared along with the certificates to them as well, so that they can be it can be used by the participants in future in their work. We are also very grateful to Dr. Paul Chegg for his time and presence today. Dr. Samar, I would like you to, sort of introduce your team, which was involved in this online course today, before we take Professor Iqbal's comment.
Yeah, thank you so much, Khazima. Let me introduce my team. We have Farkhanda with us. Farkhanda is a research officer working at our biotechnology wing, and her main focus is related to the tissue culture of orchid plants. And in addition to that, she's also working on hydroponic facilities also. Then we have Miss Miss Khatija with us. She completed her MPhil in agriculture biotechnology, and she is also working with various projects, but main are the tissue culture of orchid plants. And in addition to that, currently she is working to establish the date palm tissue culture at our facility. Then we have Mr. Rizwan with us. Rizwan has also recently completed his MPhil in agriculture biotechnology. His main focus is on rice, hybrid rice. And in addition to that, he's also working on various projects related to the tissue culture. Okay. Thank you, Khazima.
Thank you very much, Dr. Samar. And again, very thankful to all our participants as well as to you and your team who made this event a successful one. And we are also very grateful to the participants who joined us from different corners of the world with the different time zones. We're just waiting for Professor Iqbal. As soon as he joins the Zoom meeting, we will have last closing comments from him. What the fuck? So thank you very much for joining us. We just closing our today's online certificate course session. And we have also, with your permission, I would like to thank our participants as well as speakers and the organizers for this wonderful effort and conducting a successful event. For everyone's information, we had around about 120 participants who joined us for this online session today. Again, very much a lot of thank yous to Professor Dr. Paul Chegg, as well as Dr. Samar and her team at ICCBS. And with this, I request His Excellency, Professor Dr. Iqbal Chaudhry, for his closing remarks.
Thank you very much, Khazima Mazum and Professor Dr. Samar Yusuf for organizing yet another very important event on a topic which is relevant, important, and related to the food security, a technology which can be used beyond banana, and a technology which certainly has lots of merit. I'm delighted that it was, like previous program, a very popular one. 100 people have participated from different OIC countries and the other countries also, which represent that there is lots of interest. I sincerely hope that this would eventually be one of many events which you would organize in future also. And we continue to help in developing capacity in the field of modern agriculture. I have learned that there were a lot of interest, and looking into various comments, I'm impressed that there are interests which goes beyond this workshop, and there are people who are actually talking about developing their expertise by visiting places where this technology is already in practice. So This is certainly a very encouraging sign that people would like to develop know-how and technology to a level that they become expert in it. I see several messages in which people talk about master program and PhD program and internships of different kinds. And I would like to invite you to kindly visit Comstech website. It is a very happening place. There are lots of opportunities for you to visit Pakistan and other OIC countries where you would find relevant expertise. And in that particular case, if you apply for one of these fellowships, I'm sure that my colleagues in relevant section would be very happy to look into your application and wherever possible to facilitate your travel and mobility. These are online courses, of course, right now because of the situation, but certainly there would be a time very soon when we would be able to invite many of you for these events in person. In those cases, you can stay beyond the workshop for a few more weeks and learn these technologies and of course develop collaboration. So I can see a great future in this collaboration. I also see that the networking which you were able to have through this workshop would eventually benefit everyone. I'm sure that my colleagues in ICCBS and Comstech would be able to help you in future as well. Thank you very much, and thank you for being part of an exciting learning experience. Assalamu alaikum wa rahmatullah.
Thank you very much, sir. And with this, we come to end of our session today, and we hope to see all our participants again in some other activity of Comstech. But as Dr. Sub said, kindly visit our website. You will be able to find many opportunities for research fellowships and scholarships. With this, thank you very much, and hope to see you again. Bye bye and Khuda Hafiz.
Bas.